Hello everyone and welcome to Formscapes. Previously, we examined the concept of morphogenetic fields and the ways in which research pertaining to bioelectricity has come to revivify this concept within the realm of developmental biology. We saw some of the most extraordinary examples of how the influences of such fields can be empirically observed in biological experiments, and we discussed some of the ways in which these developments may necessitate the reimagining of the overall paradigm of evolutionary biology. Moving forward, we will now take a closer look at Rupert Sheldrake's theory of morphic resonance, and the implication that morphogenetic fields may be able to influence and inform one another in a non-local manner. Sheldrake tells us that the term morphic resonance is intended to imply a form of sympathetic resonance which parallels the phenomenon of energetic resonance. Energetic resonance is the process by which vibratory systems are able to transfer energy and information due to similarity of frequency. The sound waves produced by a tuning fork, for example, are able to induce vibrations within another tuning fork, given that there are sufficient harmonic relations between the two devices. Sympathetic resonance can be observed with various musical instruments such as guitars or harps. If, for example, a lower frequency string is played which shares an overtone with the fundamental frequency of a string which is tuned higher, then the higher pitched string will begin to vibrate and produce sound even though it has not itself been played. Some instrument designs take advantage of sympathetic resonance intentionally, while other instrument features are designed specifically to mitigate such resonance. A similar process is found in the functioning of radio receivers. Radio transmitters emit low-frequency electromagnetic waves within a certain bandwidth. A radio receiver which is tuned to the correct bandwidth will be sensitive to radio waves within the specified range and will respond to them, thereby receiving the information encoded within the radio waves. The radio receiver is then able to translate this encoded information into sound or visual images. Rupert Sheldrake suggests that morphic resonance may operate in a similar manner. While energetic resonance functions on the basis of similarity within a single dimension, that of frequency, morphic resonance operates on the basis of similarity within the four-dimensional space of living forms. Morphogenetic patterns which are more similar to one another exert a kind of stabilizing or regulative influence upon one another, as similar living processes are more sensitive to or more tuned into one another. Much like waves of sound or electromagnetism, self-organizing systems are constantly in states of cyclical flux. The electron orbitals of atoms vibrate, oscillating in strength with a particular frequency, which directly correlates with the energy of the electrons. Living cells, tissues, and organs are constantly in a state of metabolic flux, repeating oscillatory patterns at various levels of organization. All living organisms are complex, nested, and interwoven patterns of such cyclic processes which coalesce so as to form organic wholes. Rather than acting by a transfer of energy, as in the case of sound resonance or electromagnetic resonance, morphic resonance would instead act by reducing indeterminacy in specific ways. Sheldrake asks us to think of morphic fields as structures of probability. At the most basic level of self-organizing systems, the level of fundamental particles, we find an inextricable indeterminacy. The behavior of fundamental particles is not fully predictable, and as such, the behavior of higher-order systems such as molecules, crystals, or cells cannot be perfectly determined either. Morphogenetic fields would then operate by conditioning the likelihood of certain events so as to give shape to developing organic systems. This understanding of morphic fields as constrictions of probabilistic systems is closely related to the more general theory of causal emergence developed by the neuroscientist and philosopher of mind Eric Hoel. Hoel argues in an extremely rigorous and formal manner that macroscale structures of complex systems can actually have greater causal influence than the microscale structures which comprise them. Another way of putting this would be to say that relations between macroscale phenomena can be more determinative of outcomes than relations between microscale phenomena. A system which is composed of numerous probabilistic systems may nevertheless be reliably predicted, as the lower level events, although themselves indeterminate, average out in predictable ways due to the manners in which the lower level events are organized into higher level structures. What Sheldrake is proposing is that morphic fields could act by lowering or raising the likelihood of probabilistic micro-level events, such as the formation of a chemical compound, the folding of a protein, the division of a cell, or the firing of a neuron. This is distinct from Hoel's conception of causal emergence insofar as Hoel's theory does not require any alteration of the probabilities of micro-level events. Hoel's conjecture is rather that macroscopic structures have a causal influence which is distinct from the mere aggregate effects of microscopic constituents. Within Sheldrake's theory, it is alterations of the likelihood of probabilistic micro-level events which give shape to organic wholes. Nevertheless, there is a deep affinity between Hoel's causal emergence and Sheldrake's formative causation, insofar as both purport to demonstrate ways in which higher-level structures are able to exert an influence which is distinct from the activities of their parts, and insofar as both conceptions pertain to the relationships between the indeterminacies of micro-level phenomena and the determinative structuring of higher-level organization. As such, I think it is appropriate to regard formative causation as a kind of corollary of causal emergence. While causal emergence pertains to the ways in which higher-level structures strengthen causal relationships by imposing order upon more noisy aggregations, the formative causation of morphic resonance pertains to the manner in which such higher-level order emerges through the conditioning of such probabilistic noise. The process of morphic resonance would thus operate by a kind of automatic averaging. As formally similar systems influence one another, novel developments will come to be directed through developmental pathways, or creodes, which have been carved out, as it were, by previous systems. In order to illustrate this, we will next look at a series of experiments which Sheldrake believes may indicate the effects of morphic resonance in the behavior of rats. In 1920, the British psychologist William McDougall conducted experiments of rat behavior at Harvard University, with the intention of empirically testing the Lamarckian theory of the inheritance of acquired characteristics. McDougall's experiments involved a group of white rats, which were tasked with swimming through a tank of water and escaping the tank by one of two ramps. One ramp was brightly illuminated and thus easier for the animals to see, but this illuminated ramp would deliver an electric shock to the rats as they passed over it. The other ramp was unilluminated and thus less obvious to the animals, but passing through this exit would not deliver such an electric shock. Throughout numerous trials, McDougall's rats would eventually learn that they should only try to escape through the unilluminated exit. Although some rats would go through more than 300 trials before learning to avoid the illuminated exit, the animals would eventually learn to associate the bright illumination with the electric shock, and thus to look for a different way out of the tank. These rats would then be bred to produce a subsequent generation, which would likewise be subjected to the same tests. The breeding pairs which would produce the subsequent generation were selected before testing, so as to account for the possibility of unconscious bias on the part of the researchers, though breeding itself would be conducted after the testing of the animals. The goal here was to determine whether or not subsequent generations of rats would learn to avoid the brightly illuminated exit ramp at a faster rate than their predecessors. These experiments would continue over the course of 15 years and 32 generations of rats. In addition to the primary group of rats which were bred at random, McDougall would also conduct the same experiments upon two other groups of rats. One in which the fastest learning rats were selected for breeding, and one in which only the slowest learning rats were selected for breeding. If we presume that no inheritance of acquired learning should be at play, then we should expect that only the rats bred on the basis of faster learning should come to learn faster upon subsequent generations. The rats bred at random should come to learn at approximately the same speed from generation to generation, and the rats bred from slower learning should, if anything, come to learn more slowly on average than their predecessors. McDougall also occasionally tested a fourth group of rats whose parents had not undergone such experiments as yet another control group. What McDougall found instead was that all four groups of rats came to learn to avoid the illuminated exit at markedly faster rates from generation to generation. These experiments were then repeated by the zoologist Wilfred Auger of Melbourne University. In addition to the controls which McDougall had implemented in his experiments, Auger also systematically tested the learning of rats whose parents had not been trained in previous trials. This was to allow for a more rigorous comparison of changes in learning speed between rats whose parents had previously undergone these experiments with rats whose parents had not undergone such trials. As with McDougall's experiments, Auger found that rats of the trained breeding line came to learn faster upon subsequent generations, but also found a marked increase in learning speed for the untrained generation of rats as well. Although the results of these experiments seem to somehow be at odds with both Lamarckian and Neo-Darwinian models of inheritance, Sheldrake argues that these results align exactly with what would be predicted by the theory of morphic resonance. As the rats came to associate the illuminated exit with the correlated electric shock, the developmental pathway towards learning this association was thereby reinforced with each subsequent generation. Thus, the learning of this association came more easily not only for the offspring of rats, which had already learned this association, but also for unrelated rats, as their learning processes came to be influenced by the previous generations as well, not merely through heredity, but due to the influence of morphic resonance. As the developmental pathway, or creode, came to be carved out, all of the rats which were adequately similar to the experimental subjects found it increasingly easier to achieve this development. Morphic resonance is a theory which pertains to all self-organizing, or autopoietic systems in general. This implies that the effects of morphic resonance should be expressed not only by biological organisms, but perhaps also in molecular or even astronomical structures. For any such system, the form which is achieved by self-organization should be shaped by the morphic influence of similar systems which have developed in the past. For systems which have existed for a vast amount of time, such as hydrogen atoms, for example, the cumulative influence of the past should be so great that such systems exhibit very little morphogenetic variation. novel developments should have very little effect, as any such alterations would exert invisibly minuscule influence in comparison to the effects of innumerable past instances. Conversely, however, the effects of morphic resonance should be notably pronounced within systems which are relatively new. Synthetic structures, which have only ever been created by human artifice, therefore, should allow us to empirically observe and even measure the effects of morphic resonance. Artificial chemical compounds which have never existed previously should initially be very difficult to create. Without the influence of past forms to guide the formation of the new substance, random chance will have a greater bearing upon the formation of the substance. Formation will be less likely to occur, and the system will break down more easily until the cumulative effects of past iterations come to stabilize the morphology of the novel structure. We should expect new compounds to become more and more stable and more easily synthesized over time. Looking to the history of synthetic chemistry, Sheldrake argues that this is exactly what we find. Tyrannose, for example, is a type of sugar used as a carbon source by numerous species of bacteria and fungi, and which occurs naturally in honey. For many decades, tyrannose was considered to be a liquid, as it had only ever been observed in a liquid form. In the 1920s, however, tyrannose crystals came to form for the first recorded time, and subsequently such crystallization came to be observed throughout the world. In some cases, new chemical structures come to emerge which seem to replace previous forms. Xylitol, a kind of artificial sweetener, was first created in 1891 and was initially considered to be a liquid. In 1942, a crystalline form of xylitol with a melting point of 61 degrees Celsius came to develop. A few years later, a new crystal structure came about, now with a melting point of 94 degrees Celsius, and thereafter chemists found that they were unable to recreate the first form, perhaps indicating that the more stable structure was able to overwrite the previous, less stable structure. Within the realms of synthetic chemistry and pharmaceutical manufacturing, the emergence or displacement of new forms is a well-documented and recurrent issue. The drug ritonavir, for example, was introduced in 1996 as a treatment for AIDS. After 18 months of continual manufacturing of the substance, chemical engineers came to discover a previously unknown polymorph of the compound, one which rendered the compound significantly less effective for treatment purposes. The chemists involved found that they were unable to prevent the new polymorph from forming, and the drug had to be withdrawn from distribution. It therefore seems that, in many cases, some polymorphs form more readily than their counterparts, and the former come to outcompete the latter. As many chemists have noted, once a new crystal polymorph emerges in one location, the novel form soon comes to emerge throughout the world. Regarding this often pernicious phenomenon, the American chemist Charles P. Saylor remarked that it was, quote, as though the seeds of crystallization had been carried upon the winds from end to end of the earth, end quote. It is true that molecular crystals can act as seeds, which are able to spark a chain reaction by which the crystal formation spreads throughout a given compound. Thus, in explaining the phenomenon of new structures seeming to emerge in disparate parts of the world at the same time, chemists have sometimes suggested that such seeds travel from laboratory to laboratory upon the clothes or beards of migrant scientists who contaminate the substances of different labs and thus spread the new polymorphs. It is possible, however, that careful experiments involving novel compounds could empirically validate Sheldrake's claim that such phenomena occur due to the effects of morphic resonance. If several new synthetic crystal forms were developed, then experimenters could isolate one of the new forms and synthesize it repeatedly over a period of time, while not recreating the other forms. Then the experimenters could synthesize all of the forms involved and measure how readily the different forms come to crystallize. If Sheldrake's theory is correct, we would expect the repeatedly synthesized form to then crystallize faster than the other forms. As the theory of morphic resonance implies that novel chemical structures should become more stable over time, this suggests that we should see a measurable increase in the melting points of synthetic crystals which have only existed for a relatively short period of time. With data collected from various editions of chemist handbooks printed throughout the 20th century, Sheldrake shows that in fact there has been an often drastic increase in the melting points of relatively new synthetic crystal structures, while the melting points of naturally forming crystals has remained almost completely stable in most examples. This data is exactly what one would expect to be the case if morphic resonance were indeed at work in conditioning the behavior of these chemical compounds. Naturally forming chemicals have existed for a long enough time that the cumulative effects of morphic resonance have stabilized their melting temperatures. With synthetic chemicals, conversely, we see the effects of morphic resonance accumulating throughout the years since their creation by human beings. As resonance further solidifies the morphic fields associated with these chemicals, their melting points come to measurably increase. Some chemists have explained this phenomenon by indicating that these changes in the measurements of melting points could be instead due to improvements in the purity of these chemicals over time. Impurities in chemicals can in fact lower melting temperatures, though that explanation would not in itself account for the fact that such temperature change over time seems to have occurred primarily in synthetic chemicals, but not in naturally occurring chemicals. Improvements in purity might very well have also resulted in discrepancies over time in the measured melting points of naturally occurring chemicals as well, if the samples used were created by manufacturing. Unfortunately, we simply don't have data on the purity of the samples which were used in the measurements of those melting points, and so we can't definitively decide the issue one way or another until further tests are conducted. Again, the creation of new synthetic chemicals could rule out the possibility of these increases being due to increases in purity. Researchers could measure the melting points over time while also testing for the purity of the samples used. If a notable increase is nevertheless detected over time, then there must be some other explanation. By looking more closely at the example of chemical compounds and crystallization, we can begin to see the broader implications of Sheldrake's theory, not only for biology and chemistry, but perhaps also for our presumptions about nature more generally. Chemicals are, of course, composed of atoms. Different species of atoms bond with others in different ways, primarily due to the role of the electron shells which encase them. Electron orbitals are layers of distributed potentiality, which can contain a certain number of electrons depending upon how many shells are nested within one another. The innermost orbital shell can contain a maximum of 2 electrons. The hydrogen atom has 1 proton and thus 1 electron which accompanies it within its single orbital shell. As there are room for 2 electrons within this orbital shell, the hydrogen atom is thus able to form covalent bonds with other atoms in which electrons are shared between the outermost shells of the atoms. When a hydrogen atom bonds with another hydrogen atom, the orbital shell of each atom becomes completed, as it were, by the electron of the other atom. The orbital shells of both hydrogen atoms are then completely filled and the compound H2 is created. Covalent bonds such as this are the most common type of bond found in most chemical compounds. The most typical presumption made by most scientists is that the formation of compounds is determined entirely by unchanging laws which are derived entirely from the nature of the atoms involved. If one understood fully the nature of the atoms, then one would have a full understanding of their bonding behavior, and one would thus, in principle, be able to predict the nature of the compounds formed by these atoms, their melting points, their overall structure, and their behavior in relation to other chemical compounds. In reality, making such predictions is usually not possible. Typically, scientists do in fact have to experiment and observe in order to determine the properties of these chemical compounds. Yet the unspoken presumption remains that if these phenomena were not so complex, or if we had more detailed information, then such predictions would be possible for all chemical structures. The shortcomings of this presumption can be seen quite dramatically within the science of protein synthesis. Scientists have grappled with the protein folding problem since the 1960s, as researchers inquired as to how exactly the amino acid sequences of proteins came to determine the complex three-dimensional structures of such protein molecules. Despite decades of research, chemists are still unable to fully explain how such three-dimensional structures are dictated by the behavior and properties of the atoms which comprise them, or how protein folding is able to occur at the incredible speeds which we observe. Researchers have employed numerous computational methods in attempts to predict protein structures, though these methods have only ever achieved very limited success with very small, isolated proteins, and researchers have as yet been unable to generalize from these isolated accomplishments. In 2018, AlphaFold, an artificial intelligence system developed by Google DeepMind Laboratories, began to be employed by researchers in an attempt to predict protein structures. AlphaFold was a machine learning system which was trained by examining the known properties of over 170,000 proteins, and then used this information in order to predict protein structures. Although AlphaFold did achieve a degree of success in making these predictions, which was much higher than what had previously been accomplished, the results it produced were far from perfect and not nearly accurate enough to allow for the discovery of new pharmaceutical compounds. More importantly, however, is that although AlphaFold was able to make somewhat accurate predictions of protein structures, it remains unclear as to how exactly AlphaFold was even able to accomplish this. If some hidden laws of nature are indeed fully determinative of protein structures, AlphaFold is still not able to actually tell us what those laws are. AlphaFold predicts the structures of proteins based on data pertaining to other proteins, and although it has achieved a relatively impressive degree of statistical accuracy in doing so, it still cannot tell us why or how proteins actually fold the way that they do. What the theory of morphic resonance suggests is that we are not able to predict the structures of proteins based on the properties of their constituent atoms because the properties of isolated atoms are not fully determinative of protein structures. The higher level structures simply cannot be fully understood as derivative of lower level constituents, as higher level structures are achieved in an evolutionary rather than a strictly mechanistic manner. The protein folding problem remains a problem, not because we do not yet have the computational power needed to account for the complexity of nature, but rather because our fundamental presumptions about nature are in need of serious reconsideration. These presumptions are outgrowths of the more general, metaphysical paradigm known as mechanistic reductionism, a framework which has largely, but not entirely, dominated scientific discourse since the achievements of Galileo and Descartes. While physical science studies the actual phenomena which make up the natural world, metaphysics consists of the basic presumptions about the world which are needed in order to contextualize and direct such study of nature. Data must be interpreted within the context of scientific theories, and scientific theories must be interpreted within the context of our presumptions about what the world fundamentally is. Without such presumptions, all empirical data would simply be noise, as we would find ourselves unable to interpret any empirical data in a meaningful or consequential manner. When we turn our critical scrutiny back towards those fundamental assumptions, we begin to enter the domain of metaphysics. Mechanistic reductionism is a metaphysical framework which presumes that the phenomena we observe in nature can be understood entirely in terms of the properties found in the building blocks which make up the phenomena in question. This metaphysics is mechanistic in that, with regards to mechanical or artificial systems, this seems to be mostly true. A metal pipe is going to behave very predictably like a metal pipe, regardless as to whether it is being used as part of an engine, as part of a scaffolding fixture, or if it is being used as a walking stick. The behavior of the metal pipe is going to be due to properties which the metal pipe has in itself. The pipe does not know that it is part of a larger system, and its fundamental behavior remains the same regardless as to what machine it may form a part of. The parts rule the whole, and the whole exerts no influence upon its parts. Rupert Sheldrake's theory of morphic resonance is particularly radical in that it supposes that with regards to self-organizing or autopoietic systems, these reductionist presumptions simply do not always hold. If morphic resonance is indeed at play in the formation of chemical compounds, then a carbon atom might indeed behave differently depending on what sort of compound it is forming a part of. And this behavior cannot, even in principle, be predicted in advance even if we knew everything about the properties of carbon atoms. A carbon atom might very readily form bonds which create a compound which has existed for billions of years, and such bonds would be very secure, as the morphic fields which direct the emergence of that compound will have been solidified by the cumulative influence of morphic resonance which have accrued over time. Sheldrake suggests that morphic fields may exist at all levels of natural self-organization, not just within the bioelectric distributions of biological organisms. The forces which hold a single atom together and govern the behavior of electron orbitals could themselves be forms of morphogenetic fields which have concretized over the course of billions of years, such that they seem to behave with law-like consistency. Once an atom begins to bond with other atoms to form chemical compounds, higher-order morphic fields begin to emerge. As similar chemical forms come to resonate with one another, more consistent patterns of self-organization become established through morphic resonance. The lower-level morphic fields of individual atoms then come to be interwoven within the higher-order morphic fields of chemical compounds, and the morphic fields of chemical compounds then become interwoven within the morphic fields of macromolecules, such as proteins. As Sheldrake indicates, the theory of morphic resonance is fundamentally a theory of formative rather than mechanistic causation, one in which patterns of self-organization are achieved through the creative advance of the natural world rather than being predetermined in advance by transcendental laws which are simply given for the basic building blocks of the world. Thus, Sheldrake's theory finds itself so at odds with the prevailing paradigm of academic science in that it is representative of a kind of thinking which is process-relational rather than mechanistic. Like the metaphysics of mechanistic reductionism, the paradigm underlying Sheldrake's thought is in essence Neoplatonic, but it is a Neoplatonism of evolutionary development, rather than of static timelessness. In the next episode, we will continue our discussion of the metaphysical implications of morphic resonance theory, exploring how this theory relates to the work of process philosophers such as Henri Bergson and Alfred North Whitehead, and look at how we might understand morphic resonance in terms of the philosophy of science. We will also begin to dive into much more speculative territory, as we look at the broader implications which morphic resonance might have for our understandings of supposed parapsychological phenomena, as well as our understandings of ritualistic or shamanic practices found throughout human societies. And that's going to be a wrap yet again. Hope you like this episode, and if you want to see more, remember to like and subscribe. I'd be very much appreciative of that. I know these videos are pretty long-winded, so if you're making it through these things, then pat yourself on the back. I know we all have other things we could be doing, so I'm trying to make sure that these are as worthwhile as possible. But until next time, thanks for watching.